All right, ladies and gentlemen, for our second biochemistry presentation, we're going to discuss the organic macromolecule of carbohydrates. Now, you'll notice even in this name, there's like pieces of words. So if you see right here, carbo is related to the word carbon, and hydrates is, hopefully you realize, sounds a lot like water. And so it's related to water, which has the chemical formula of H2O. And so this whole idea of CH2O is going to actually be a really important concept later on. So let's start with some of the basics. First off, carbohydrates have a more technical name of saccharides. And saccharide means sugar. So you can refer to a carbohydrate in a multitude of ways. You could say it's a monosaccharide, which means that it's one sugar. And monosaccharides, this one sugar unit, these are the monomers for carbohydrates. And then you can also have disaccharides, which is two sugars, because that's what the prefix di means. Or you could have polysaccharides, which means many sugars, because that's what the prefix poly means. Disaccharides and polysaccharides are examples of the polymers for monosaccharides. Or I'm sorry, the polymers for carbohydrates, for saccharides. So the monosaccharides, which are the monomer unit, actually there's different monomer types. So this one right here is actually the monomer glucose. All right. And you can see that there is carbon, there is hydrogen, there is oxygen, and that's it. That's all there ever is in carbohydrates. Down here you can see this one is the carbohydrate monomer fructose. And what's important here is that you'll notice again, just carbon, just hydrogen, just oxygen. But they have slightly different shapes. Now, we mentioned in our previous presentation that one of the key ideas in biology is going to be that structure equals function. So if something has different shapes, it can have different properties. And living organisms actually do use glucose and fructose slightly differently. They do the same thing with them, but they go about it in different ways, and that's because they have different shapes and therefore different properties. Now this one right here you can see is already labeled, is galactose, which is another simple sugar. And you'll notice again, carbon, hydrogen, oxygen, and nothing else. In fact, what you will notice is that if you look at each of these carefully, each carbon, for every one carbon, there is always two hydrogens and one oxygen. They're always in the same ratio. So that if I have, let's say, 6 carbons, since there's always one carbon for every 2 hydrogen, I'm going to have 12 hydrogen. And since there's one carbon for every 2 hydrogen for 1 oxygen, I would have 6 oxygens. If, again, I wanted to go about this differently, and say I had 5 carbons, since it's 1 to 2, I would have 10 hydrogens, and since it's 2 to 1, I would have 5 oxygens. And that is going to be consistent for the monomers. Now, what I want to make sure you grasp here is that there is more than one monomer type. So if I wanted to create a disaccharide, I could connect glucose and fructose. And that's 2 monomers becoming a new polymer. If I wanted to create a disaccharide, I could connect glucose right here and galactose right here. And that will be a different polymer than the first one. And that's really important, that because there's different monomers, this increases the variety of polymers. Okay? So let's talk a little bit about some of those polymers. So I want to start with disaccharides. Now, disaccharides are very simple polymers. As we said before, they have the prefix di, which means that they have 2 monomer units connected. This is about as simple a polymer as you can get. So right here, you have glucose connected to fructose. And you can see here that they're bonded around one of the oxygens. Now, glucose and fructose connected is a disaccharide known as sucrose. Now, you're very familiar with sucrose because this is what we know as table sugar. So your table sugar is actually not a monomer, it's a polymer of carbohydrates. And your body breaks it down into glucose and fructose and then uses the glucose and fructose. Down here, you have the monomer galactose and the monomer glucose, again connected. And you'll notice the connections are the same. And that's really important because I said in the previous presentation, the process that connects them is the same for every polymer. The process that breaks them down is the same for every polymer. And so down here, galactose and glucose connected together, this is actually what we call milk sugar, or what's known as lactose. Some people you know might be lactose intolerant, and that means that they are unable to break down this bond. They don't have the enzyme necessary to break that bond in two. And so when the galactose and the glucose get into their digestive system, it causes like cramping and gas and can cause diarrhea and actually can make them very ill because their body can't break it down appropriately. So these two are examples of what happens when you connect the monomers together, because they're not the same as each individual piece. Like when you bring them together, it's something new that your body reacts to, or that any living organism will react to in a different way. And that's an important idea. Now, the thing is that disaccharides are not the only polymer that can be made. Living organisms can also make very long and complex polymers. And these are known as starch, which you find in plants, cellulose, which you also find in plants, and then there's a third one known as glycogen, which you find in animals. Now, these all are carbohydrate polymers, which means they will have many monomers. Now, for the majority, starch, cellulose, and glycogen are glucose-based. So we're using this one monomer. But the thing is, if you take a look at glucose, see here how it has a very specific structure? You can actually, like, flip it over. Imagine a coin. One end of the coin is heads, one end of the coin is tails. So while for the most part the coin's very similar, heads up, tails up is actually a slight difference. So when it comes to these polymers, whether you connect them heads, tails, let's say heads, then heads, then heads, then heads, then heads, so that every time it's heads up, or if you connect it so it's heads, then tails, then heads, then tails, then heads, then tails, or over here, they're all heads up, but you're connecting them in weird, complicated chains. And that's the thing about polymers. The connection is always done with the same enzyme and the same process, but the connection can be done in different ways. So here you have starch. Starch is a plant product. You eat it a lot in things like corn, potatoes. Starch is a useful source of energy. Your body actually breaks every one of these connections between glucose molecules to get energy out of them. But starch is connected in such a way so that every glucose molecule is assembled facing the exact same way. So in our example, that means heads up, heads up, heads up, heads up, heads up. Now here you have cellulose. Cellulose is also in plants. Cellulose is what makes up plant cell walls. Cellulose is what you cannot digest, which is in fact why corn is notorious for you being able to know when you have eaten it. because your body cannot digest parts of it. In fact, cellulose is a key component for fiber because it allows parts of your digestive system to work in ways they normally wouldn't, and that helps keep you healthy. But the difference between cellulose and starch is that cellulose, and you can see this right here with these little structures off the side, is assembled so that glucose is in one way, glucose is in the opposite way. Glucose is in one way, glucose is in the opposite way. So again, using our analogy, where starch is heads, 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 cellulose is heads, tails, heads, tails, heads, tails. And if you recall, since structure gives things its properties, this will have different properties than that. And the chemicals in our body cannot break down cellulose. And so it moves through our digestive system undigested. Now, glycogen is how animals store sugar. And again, using our analogy, it's always heads, 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 heads. But the difference is starch is a straight line. Cellulose is a straight line. And glycogen is branching. And you can see that here and here and here. And the reason for that, because it takes up less space. Animals don't have a lot of storage space because animals need to move. And since they need to be mobile, they can't be carrying around just everything they need. Plants, particularly like trees, can just be gigantic. So they can just have all the storage space they need. Animals, however, do not. And so animals actually store glycogen in the liver. We don't store a lot, and it frequently gets used and then replaced and then used and then replaced. But that's what we do. We branch it so it's easier to store. So here you have 3 different types of large polymers of carbohydrates. So we've talked a little bit about the structures in carbohydrates. So what we want to discuss now are the uses of carbohydrates. Now, more than anything else, this is the single most important thing about carbohydrates. Carbohydrates are the primary energy source in living things. Carbohydrates are where every living thing, animals, plants, bacteria, humans, all of us get the most of their energy. Carbohydrates also have some structural uses. For instance, the macromolecule polymer cellulose, this structure right here, actually is the biggest component of cell walls in plants. And there are also, different branching carbohydrate chains, different polymers that a lot of cells use as identification, which is something we'll discuss later on when we talk about cell reproduction and the cell membrane. All right, so those are the basics of what carbohydrates are, what their monomer is, what their polymer is, and what they are used for. Thank you very much.